Good day, everyone. Today I'm going to start the unit 2, which is basically the selection methods. In this, we are going to look at the importance of the selection methods in case of silkworms, the kind of characters that we are looking at, and how are new breeds going to be formed, and how are new breeds authorized to be formed. So you are going to see individual selection, mass selection, then different types of breeding, as well as the formation of the new breeds. So this germplasm that has been there, like we have already seen around 432 base pairs, this is there. And there's an international consortium of Bombyx mori, in fact. And there are nearly 3,000 types of Bombyx mori that is present. That is your silkworm basic one. So we have univoltine, bivoltine, polyvoltine breeds, and univoltine and bivoltine, meaning as such, you have the breeding that takes place annually is univoltine, bivoltine is twice, polyvoltine is many times in the life cycle. So, because of which you have the quality of silk that is going to be differing based on the fact or, based on the kind of times you have silk being produced or based on the life cycle. So you have the geographical races also, which contribute to your genetic stock, in fact, and there are a lot of mutations. I'm just repeating or I'm recapitulating what has been taught yesterday. So we'll go to the next page. So what are we looking at in terms of individual or mass selection? We are going to look at the qualitative characters. This has also been dealt with yesterday. So we have seen the egg color, the chorion color, and the kind of markings that are there on the larva, the cocoon's shape, color, size, nature. Is it grainy? Is it medium, fine, coarse, or flossy? In fact, and all of this has been manipulated and has been experimented with in your Bulgarian characterization basically. Now, when it comes to the qualitative or the characteristics, we have seen all of these. When it comes to the quantitative characteristics, we are going to look at the hatchability, the larval duration during 1 hour, and the pupal stages or the instar stages, pupation rate in terms of percentage, the number of normal eggs that are being laid, weight of the normal eggs that are being laid in the in grams, and then you're going to see what is the abnormality percentage, number of normal eggs per 1 grain, and then you're also going to see the fresh cocoon grades in percentage. That is, immediately after the cocoon is harvested, the floss is removed. Cocoons are going to be graded according to the kind of pupa that are alive and that are present. If at all there are double cocoons, meaning are there two insects, are there two silkworms, and how many of the cocoons cannot have the silk thread extracted from them. So all this sorted is sorting is done, and then they are going to be sorted out into these three characteristics, and then they're going to be weighed. And also you have the fresh cocoon weight and the shell weight, the shell percentage, and then how many cocoons are present as per or in one box of eggs, that is around 20,000 eggs in a kilogram. And the filament length in the meters, that is your silk filament, in how many meters is going to come. So one revolution on the approved, which is your the quantitative measure which is used to measure the silk thread. It's around 1.125 meters, or around 400 revolutions on the approved is around 450 meters of your filament, silk filament. And then what is the weight of the silk filament? After the cocoon is reeled, what is the dried weight and how much of it is constant? And size, again, you're going to calculate it by filament weight in grams by filament length in meters into 9,000. 450 meters into double, basically into 2, you're going to do. Reelability in percentage, again calculated by reeled cocoon number by number of ends that are being fed into 100. And what is the raw silk that is going to come? Percentage. That is again going to be a filament weight by dry cocoon weight into 100. And what is the amount of raw silk that is being yielded by one box of silkworm eggs? So you have a formula for that. Fresh cocoon yield from one box of eggs into what is the good quality that is there. So good quality cocoons percentage into 0.01 into percentage of dried cocoons obtained from the fresh cocoons after drying to the constant weight into 0.01. So this 0.01, all of this is constant. So you this dried cocoon weight you're going to multiply into again raw silk percentage into zero point 0.1 again. So all this 0.1 is constant. So with this you're going to get the raw silk that is going to be yielded from one box of the silkworm eggs. And then we are going to see according to the consumption of the mulberry leaves how much of it is being utilized. So consumption index. So this consumption index is again dry weight of the mulberry leaf ingested in milligrams during the feeding period by feeding period duration in the days into average dry weight of one larva during the feeding period in milligrams. So dry weight of mulberry leaf ingested in milligrams and by the feeding period duration in days into average dry weight of one larva during the feeding period in milligrams. Now we also have the growth rate. This growth rate is the body weight, body gain during the feeding period by the feeding period duration in days into average dry weight of one larva during the feeding period in milligrams. So this gives you the growth rate of one particular cocoon or the silkworm basically. And then how much of the mulberry leaf is ingested. So this is your leaf ingestibility. That is equal to again the dry weight of the mulberry leaf ingested in milligrams by the mulberry leaf supplied in milligrams, the dry weight of it as such. So the dry weight of the mulberry leaf supplied in milligrams into 100. It's a percentage. And then you have the food digestibility. Now it might eat so much or so many milligrams of mulberry leaves, but how much of it is being ingested, how much of it is egested, so that is your digestibility. So when you see this, this is AD, food digestibility is equal to dry weight of the mulberry leaf ingested in milligrams minus dry weight of the excrements. You're going to measure or weigh the excrements in milligrams. So, dry weight of the mulberry leaf ingested in milligrams by minus the dry weight of the excrements in milligrams by total dry weight of the mulberry leaf ingested in the milligrams into 100. Now, how efficient is your digestion ingestion? So, this is going to be efficiency for conversion of the food supplied in the products. So, this is going to be ECS. So, that is your dry weight of the products obtained, namely body gain during the feeding period or the cocoon shell or the pupa or the eggs by the mulberry leaf supplied in milligrams, that dry weight into 100. Now, efficiency of conversion of the food ingested in the products, that is ECI. So, food ingested, food supplied is one thing. You how much amount of mulberry leaves, dried ones you're giving, and how much of the body gain is seen. But when it comes to food ingested, how much of it is being efficiently converted. That is equal to your ECI, which is equal to your dry weight of the products obtained. That is your body gain again during the feeding period. So you're going to check the weight of the cocoon or the pupa or the eggs per or by dry weight of the mulberry leaf ingested in milligrams during the feeding period into 100. Sure. And good evening, sir. Yeah, sir. Pranatini. సార్ అదేంటి లైవ్ స్ట్రీమింగ్ ఆగిపోయింది. యాక్చువల్లీ బిఆర్ అంబేద్కర్ కేమ్ ఇన్ టు లైన్ అన్నమాట. సరే సెషన్ స్టార్ట్ చేస్తే మళ్ళీ అది కాస్త వాళ్ళు ఐ థింక్ దర్స్ నెట్ ప్రాబ్లమ్. ఐమ్ నాట్ రియలీ షూర్. సో 10 minutes into the క్లాస్ మళ్ళీ వాళ్ళు ఏమీ లేరు. ఒక్కసారి కెన్ యూ జస్ట్ హ్యావ్ లుక్ ఇన్ టు దట్? ఓకే సార్. షూర్ సార్, షూర్. ఓకే సార్, థ్యాంక్ యూ. All right. So I was here near the fresh cocoon grades in percentage. We have already seen basically the qualitative methods. Now we are looking at the quantitative methods. Like I have already told you, in this we are looking at the efficiency of conversion of the food digested in the products, which is your ECD, which is equal to your dry weight of the products obtained. That is your basic body weight gain during the feeding period, or the cocoon shell or the pupa of the eggs, or the dry weight of the mulberry leaf ingested in milligrams, minus the excreta, that is the dry weight of the excreta that is there in milligrams into 100. Now, how are we going to fix the characters, or what we are going to see? Look at the different kinds of silkworm sources and how we are going to characterize them. So most of these silkworm accessions have a green or grayish egg serosa, serum color, white or yellow chorion color, plain larvae or marked larvae. And then you're going to have a constriction at the end or basic oval shaped cocoon. So the breeds in white, that is the cocoons, are usually the dominant ones which are mostly available. And all of this data is regarding or like, during the pure lines which is there, they vary from the 180th hour in the breed Gargana 2 to 218 hour in the breed AS. So that is the kind of variation that we are going to see over here. Now, most of the silkworm accessions, you're going to see that, they have a high hatchability rate. That is, most of them are able to hatch into the progeny. 80% of the silkworm accessions have a 85% or higher pupation rates. So around 26% of the strains have around pupation rate, that is formation of the pupa from around 85 to 90%. Around 39% have around 90 to 95%. And 15% of the breeds that are there, very good ones, have a pupation rate, that is formation of pupa, rate higher than that of the, higher than that of 95%. So we need optimal conditions for such kind of pupation rate to be obtained. So again, based on this, you have different kinds of cocoon weight also, which is around 1.8 to 2 grams. 2, this is around 43%. 35% of the cocoon weight is higher than that of the 2 grams, which is like very good. And if you see, many races have even, weight of the shell more than that of the 0.35 grams. So this is also a very good rate again. So you have around one box of eggs having a yield of more than 25 kilos. And this is seen in more than 93% of the silkworms. Now, most of the races are having around, that is around 37%. You have a cocoon yield from around 30 to 35%. 29% of them have around a cocoon yield of 35 to 40 kilos, which is very good again. And 5%, the top quality silk, you have around 40 to 45 kilos. And then you have 50% of the accessions having a filament length higher than 900 meters. 17% having around 900 to 1000 meters, 21% around 1000 meters, that is around 1 kilometer of silk filament you're going to get from so much percentage of the silk cocoons, in fact. That is your reliability. So around 31 accessions were tested by Vasileva and Senov in the year 2001. And from their resources, you could see the different kind of development that has been seen. That is your parthenogenetic development. And the results show that the highest parthenogenetic eggs hatchability, that's around 20%, was detected in the races SHV, TV, HESA2, Syria2, Mysore 1.70 to 4 to MNB and China. So these are the different kinds of races in the silkworm breeds. Fecundity, that is your ability to hatch the eggs. So this is again going to be given by or the highly productive silkworm races. You can see that in the Japanese type races that are there, and then you can get to see the moth emergence percentages and the different kinds of races that are there, and the percentages have been given in your textbook. You can just have a look at it. But then you see that in Japanese type races, the amount of food ingested was the highest and lowest in Hebar. In Chinese type again, the food ingested was the highest. In Radza, that is your Bulgarian race, is around 54, that is around 3.784 grams. And lowest in Hebar 2, which is another type of race. So again, food ingested digestibility in different kinds of races has been given, especially in polyvoltine races. So it's around 0.94, which is your food ingested. Digested is around 0.34. So this is the kind of data that's always that's been produced. And the leaf ingestibility was the highest in the Chinese type race, and highest food digestibility, the ability to convert whatever dried mulberry leaves to your so that it reflects in the body weight gain has been seen in the tropical races. That is your super Bratza, and food digestibility was lowest in your Hebar 2, KS, and Merefa. So consumption index was also tested, and you see that Radza is around 0.8 to 9 milligrams, and you have different kinds of other races also who have high leaf ingestion and consumption index. So polyglutton race combined with the high consumption index with the low food intake. So because of which you have the interracial differences, they are going to be negligible as such. Now, you see that the polyvoltaic races manifested very high growth rate. So, the efficiency of the food that has been converted into your body weight gain was the highest in Super 1, Merefa, Ratsa, and lowest in Hebar 2 and Bondi 517. Now, all this food has to be utilized so that, proper or good quality silk is produced, silk filament. So the utilization of the food supplied for the silk shell was highest in Super 1, Ratsa, and the lowest in Hebar 2. So utilization of the food supplied for the eggs was the highest in Super 1, Hesa 2, and lowest in AS and Hebar 2. Now this is again mostly, showing the efficiency of the food digested, ingested. I mean, sorry, it should be the other way around. It is food ingested and digested. Then it should be manifested in that so that, the races you're going to see high leaf ingestion and digestion. They showed that lower food utilization and vice versa. So when you see the hybrid vigor and the heterosis expression in the first filial, that is your first progeny F1 was low or either negative. And when you come to see the different kinds of characterization at a molecular level at the of the silkworm germplasm, then you get to see that in the gene pool, you have different RFLP markers, you have your mini DNA markers, microsatellite markers, and RAPD markers, and different kinds of AFL, AFLP, that is your amplified fragment length polymorphic markers. These are going to identify the duplicate accessions that are present, and then according to the different kinds of traits, you can map and at the same time tag molecularly. So this molecular characterization of the particular genes or your DNA is going to help in identify the genetic distinctness of a particular race or a breed or stock. So what will happen is this will help eliminate the duplicates, because of which your plasm maintenance is going to come down, the cost of it as such, and the volume of work involved in getting better breeds or in getting proper lines is going to be less. So molecular markers again linked to your metric and resistant traits. So subsequently mapping the silkworm gene is very much essential. Also the genetic markers, if you're going to see if there is any kind of heterogeneity or heterozygosity, if at all, you apply the markers, then you can actually see the changes if at all they're there. So it is a routine procedure to apply the molecular markers to the silkworm germplasm. Now, how are new breeds formed? So the first step is evaluation of the initial breeding or the breeds that are there. How is it done? Based on your physical traits, that is your phenotypic evaluation of the main qualitative and the quantitative breeding characters. Then what are the variants present in the phenotypes? And then you're going to see the value of the quantitative characters. Then how heritable are these quantitative characters? And then what is the relation between the different quantitative breeding characters which are there? And is there any kind of regression between the main quantitative breeding characters present? So for the breeding new silkworms, you have different methods that are being used. First is segregation from the foreign F1, that is first filial hybrids. So in Bulgaria breeding station, this was done, wherein you have selection. Most of the older silkworm breeds like Radza 1, Radza 2, 3, 4, 5, and 6. So what is it that you're going to do over here? So the initial population, say for example, the Japanese F1 hybrid is taken into J124. Once this is done, breeding, selection of larvae that have markings and that have elongated cocoons in the second filial generation. So you're breeding J124 into with C122, which is a Japanese F1 hybrid, and then you're going to select the larvae which are having their distinct markings and elongated cocoons, and then you're going to take, this is based on the phenotype, you're going to see the elongated shape, you're going to see the larval marking. So you're taking it based on the, or you're evaluating, or you're selecting based on the phenotypes. Then you're going to mate the selected moths. Then batches are reared. Again, you're going to select based on larval markings, cocoon shape, texture, and other kinds of quantitative characters for the 11th to the 16th generations. In this manner, you got a new breed which is known as Radza-1. And then you're going to separate from the foreign F1 hybrid in the manner that KK is a pure line that was selected in the Bulgaria, which is a common first filial hybrid that is formed from the pure mother line. But that was because of some kind of a mistake that was seen in the sex determination, because of which what happened was mating within the pure line resulted in a formation of KK. So you have the initial South Korean hybrid, and again selection based on the phenotypical characters such as the constricted cocoon, and then mate for 4 generations, and then rear the batch, then select again based on the cocoon shape and other quantitative characters, and perform auto breeding, breeding within the same line. So that is going to be performed for nearly 9 generations. And again, you're going to select based on the cocoon shape and other quantitative characters through which you got your KK, which is the new breed that was formed. Now, Japanese races also have been selected. Initial population is J124, and based on the initial layings of the eggs, especially the 6 layings, and then future sublines, keeping them in mind, selection was performed, inbreeding was done. Again, phenotypical characters are involved, and then you're going to check the ability of each line. That is, each individual race or line of this particular J124 sublines, and cross them with the Chinese subtype 157K. So, and see whether there is any kind of combining ability and hybrid vigor present. Again, once this is done, you're going to check it over there for your combining ability. Once this is performed, leave it at that. Perform inbreeding within the sublines for another 7 to 8 generations. Again, check the based on the phenotypical characters such as larval marking, cocoon shape, texture, and other quantitative characteristics, and also the combining ability of each line with other Chinese types. Once this is done, you have the best specific combining ability, so that is around super 111, through which you got a new race, which is your super 1/11. This is ready for direct hybridization again. Similarly, you have the Kinshu, through which 3 sublines were selected and inbred, and like out of which 2 sublines were rejected, and then again 1 subline which is there inbreeding was done for around 3 generations. Again, based on the phenotypical characters, selection was done, and then you had a new breed formed, which is the Hebar 1/18. In this way, breeding methods was used. Please give me a moment. In this way, the breeding method, it's been formed that all the 4 lines were performed or found in the same manner. So initial population was cross between female Naig 1 and also between males of KK. So, and then back crosses for many generations together. Finally, you have a new silk line, EVA 1, that is being read. Now, when we look at the selection that was made with individuals of EVA 1, then you're going to again cross it using selected males of KK, because of which pure basic line KK was maintained in the big volume. Sorry. Right. So, because of which, what happened is you have the EVA one, which got maintained in lower volumes, different kinds of, like, less number of batches that were there, and many times backcross was performed, because of which you have the four new silkworm sex limited for the larval markings that was formed, and they have pure lines having similar qualitative and quantitative characters, and these are valued according to those in the original lines, and they are widely adopted in the field, and especially in the commercial productions. So these are the HESA 1 KK, Gargana 2, along crossed with Westlets 2. Now, how is it that you're going to get a transgenic silkworm? So, Japanese scientist Tamura in the year 2000 succeeded in producing a transgenic silkworm in the NIS Institute. So, what happened? Tungsten needle was taken along with a glass capillary, and in the, what exactly was done was micro injection into the eggs. What was injected? It was a transposon DNA vector along with the enzyme, which is your transposase. Then genome editing was done by what your talents or your zinc fingers, that is your mRNA. You have the enzymes caspase or Cas9 nucleases, which is again mRNA or protein, which is going to be involved along with your single guided RNA. And then you have the donor DNA, which is there, because of which the gene was knocked in, because of which what happened was a hatch of mosaic GO animals was produced, and then this mosaic adults were crossed together and then checked for according to the markers, screening of the first generation or the next generation by the different kinds of transgenic markers, and then you can see the DNA amplified using the PCR method, because of which you could find out whether the gene has been amplified or not, or the presence of gene also is confirmed. Now, who does all this authorization? Who gives permission to perform such kind of experiments? It's the races were developed by CSIR and the TI, which is present in the Mysore. We know that, Karnataka is one of the silk capital of India, in fact, and you have a lot of silk. So KSIC, that is your Karnataka silk, is in fact, um, controlled by the government of Karnataka. So they have different kinds of, it's a government organization that is controlling your silk, especially in the case of your Mysore silk and Karnataka. So you have Shinichi, traditional polyvoltane hybrids such as the pure Mysore or into Shinichi, and then traditional polyvoltane crossed with your bivoltane hybrids, which this gives rise to your pure Mysore into NBD4D2, which is another type of breed or race. So many types of polyvoltane breeds and hybrids of the past were actually tried, and you have different kinds of so that what you call it, your polyvoltane breeds are improved. So because of which you have a lot of improvement in the polyvoltane breeds as of now, with the production of Kolar Gold, Kollegal Jawan, Mysore Princess, Hosa Mysore. All of these are different kinds of polyvoltane breeds that are present basically. Now, these crossbreeds, whatever are there, if you actually do the particular breeding, the exploitation or the ability that they can produce silk of a different type was exploited from as early as 1920. But it took quite a lot of time, and it was only after 50 more years that improved rearing technology actually gave rise to the introduction or the crossing of polyvoltane breeds with bivoltane breeds, hybrids, using the bivoltane breeds came into existence. So, though 1920s, they understood that could be done. It was only after 50 years, that is in the 1970s, that they could cross the polyvoltine along with the bivoltine hybrids. So this resulted in a linear improvement of the cochineal silk content, as well as the byproducts, or the it was, that is known as the rendita, has come down. In the year after 1970s, in the year during the 80s, different types of new breeding approaches were seen or tried with they were trying to find out whether there was a short larval duration, and whether they in the polyvoltines itself, whether there was a higher silk content, and whether they could breed better quality of silk, because of which new breeds like MY1, RD1, P2D1 were developed. Then these were again crossed with bivoltines. So you have NB1B and NB4D2 as the male components. Three hybrids were developed, and this was authorized in 1995. So you have different kinds of polyvoltine races today, which is your MY1 into NB1B, RD1 into NB18, P2D1X into NB1B. So what happened was, after this, productive polyvoltine and bivoltine hybrids were seen or produced during the 90s. So I'm just basically telling you the evolution of polyvoltine and different kinds of hybrid races that took place over the time, especially in India. So you have breeds like BL23, BL21 that got evolved, and then again in different areas as such. So you have BL43 and B4D2, that is your Kapila, with better productivity and quantitative traits than compared to your PM into 4 NB4D2. They were identified, and all of this was authorized by your Central Silk Board for commercial exploitation in the year 2002, that is. And different areas, again different kinds of what do you call it, experiments were done as such. So irrigated area, non-irrigated area. So you have a highly productive polyvoltine breed BL67 that has a better productivity quality traits as compared to the the other breed, which is your pure Mysore, that was developed. And then new breeds were crossed again with bivoltines. So you have a new polyvoltine bivoltine hybrid that was produced. That has a better productivity trait, higher silk recovery, and it is known as an A grade silk. That is by crossing Kaveri, which is that is your Kaveri basically. That is the hybrid that was formed by crossing your BL67 into CSR 101, which is BL67 is a polyvoltine, CSR 101 is a bivoltine. So you have a Kaveri silk, which is an A grade silk. And this hybrid is tested on a large scale, especially in Karnataka, Tamil Nadu, and AP. So the highest of this hybrid recorded an average cocoon yield of 54 kilos per 100 DFLs, and this was your cocoons fetched a higher rate of 15 to 20 per kilo with an A grade silk certificate as compared to the PM and crossed with your NB4D2 type. That is your Kapila. So again, this is there in the irrigated zone. In the rain-fed zone, you get to see that the existing PM and C Nichi that has been in the rain-fed area has been replaced with new polyvoltine and polyvoltine hybrid. So you have a Varuna. Like I told you, rain-fed zone. So Varuna is the species. 8L24 into C Nichi. This is the breed that is there. So it has a high survival, better productivity trait. And average cocoon yield is around 31 kilos per 100 DFLs. Rendita is around 10 to 11 as compared to the 23 kilos and 12 to 13 rendita realized from the popular hybrid. So your productivity has been more. When it comes to bivoltine breeds and hybrids of the past, 70s time you have breeds like NB7, NB18, NB4D2, etc. that were there. That was seen in Mysore, Karnataka, KB at Kalimpong, Northeast, and SH6YS3 at Dehradun, Central North. And you have what do you call it, C110, C108 at Kashmir, which is an exotic. You have Kashmir silk also. This is an exotic collection that was evolved. Now we have bivoltant hybrids as well. Got over. Bivoltant hybrids crossed with pure bivoltants crossed with bivoltant hybrids like Ka into NN60 and other types were highly found to be very good initially, but then significantly it did not make much of a difference as compared to your polyvoltant bivoltant hybrids that were there, because they were much more qualitatively and quantitatively better as compared to bivoltant crossed with bivoltant hybrids. And this is one thing. And then when it comes to the new productive bivoltant breeds that were seen, you get to see that whatever that were developed earlier, like in the case of your bivoltant breeds, the shell ratio realized at the commercial level was only around 18 to 19%. This is only slightly higher than the crossbreeds that were there. So again, you had an Indo-Japanese collaborative project, JICA, in which they were trying to evolve the hybrids having a higher survival rate and higher cocoon shell ratio. So that is around about 23%. So this was coupled with better reeling traits. As a result, five hybrids, that is CSR2, CSR4, cross was there. CSR2, CSR5, CSR3, and CSR6, CSR12, CSR6, and CSR16 and CSR17 were crossed with each other, and they were identified as highly productive hybrids and were authorized by the CSB or Central Silk Board for commercial exploitation. But this was around the 90s, 91 to 1999. So this is one thing. And then when it comes to the hybrid CSR2, CSR4, and then CSR2, CSR5, you have shell ratio around more than 23%. Raw silk is around 19 to 20%. And they are being exploited commercially on a very large scale by the farmers, especially during the months from September till February. And then again, they have a rendita on an average of around 6 produced. The quality of the silk is around 2A to 4A grade only. So these again have, there were again three productive hybrids that were there: CSR3, CSR6, CSR12, CSR6, CSR16, CSR17. So what happened was they tried to do reciprocal crosses along with them. So the raw silk percentage is around 18 to 20%, but you're going to get a 2A, 3A grade silk, not your A grade silk as seen in your Karnataka. So commercial exploitation was performed again during favorable months. So if you see these kind of breeds, that is CSR3, CSR6, CSR16, 17, were having an average cocoon yield of around 65 kilos per 100 DFLs. Now, if you see bivoltant breeds and hybrids, you get to see that depending upon the climatic conditions, the conducibility of the silkworm rearing is also going to change. So it is not going to be very, very useful if at all, you're going to rear high voltage high yielding bivoltant breeds during the summer season. So based on this, again, we need something to, to rear during the unfavorable season. So because of which CSR 18, CSR 19 were evolved. So what happened was, using robust breeds, the hybrid combination CSR 18 and CSR 19 was developed, and this was again authorized by the Central Silk Board in 98. And the hybrid recorded were the average yield of around 50 kilos per 100 DFLs during the non-productive, that is the summer months. And as a special 10-year productive bivoltant breeds and hybrids were seen. So what were they trying to do? There, that is the directional breeding, so that you get a better productive bivoltant breed. We have bivoltant versus polyvoltant yielding extremely well silk, A grade silk. But if you want only bivoltant, what is it that you're to see? So you're going to take around CSR 48 with having a longer filament length more than 1500 meters, thin denier, your thickness, and then by utilizing the thin denier breed, you're going to get a productive hybrid having a filament length of more than 1400 with thin denier of less than 2.4. So thin denier hybrid again CSR 48 and CSR 4 has been formed in this way. So no difference in the yield has been seen basically, and it is a good raw material for manufacture of your zari, chiffon and georgette fabrics. So they're all different types of silk now. Now, when it comes to the sex limited breeds, that is, they are going to be pertained or restricted to either the male or the female. So, that is a boon for the egg producers. So, certain drawbacks are also there. So, two new sex limited breeds, that is your CSR 8 SL and Nandi CSR 2 SL, have been developed. So, what happens is you're going to get a yellow colored female cocoon, white colored male cocoon. So easy to identify both the male and the female even before they hatch. So this is one thing which is extremely helpful for the graiнаges, because of which you can easily separate the male and the female cocoons, saves a lot of time, saves a lot of labor and money, and you do not have to wait for the cocoons to be broken or to go into the pupal form for identification between males and females. Similarly, in place of the normal CSR2 as a male component with PM, new breed Nandi having a sex-limited cocoon color was developed so that you have the yellow cocoon female, white cocoon male. Now, double hybrid for high egg recovery and cocoon crop stability. So, because of this kind of high cocoon shell ratio, low pupal rates or low pupation rates in pure races, you need more care, you need more attention to handle the pure races. So you need better or effective labor, which is again very costly. So keeping this in mind, you have a double hybrid, which is CSR 6, CSR 26, crossed with. The progeny is then crossed with CSR 2, CSR 27. So it's a double hybrid having four distinct lines in it. So it has been selected for the commercialization. So when it comes to the bivoltant breed and hybrid IBR, you have suboptimal conditions also which are there basically. So again, when you kind of see or by introgression the amylase genes from this particular, they are the polyvoltant breeds basically. So pure maestro nistari into CSR2 and CSR5 two breeds namely. So you have evolution of two more breeds, which is Gen 3 and Gen 2. Now these were again crossed. So Gen 3 and Gen 2 with the higher amylase activity. Amylase is necessary for breaking for the next stages basically, because of which higher amylase activity has been developed. So in case when you see the summarization of this particular lesson, you see that there's a lot of germplasm in different kinds of place, but you have countries in the world, and you have more than 4,000 silkworm germplasm accessions available from different countries. But we also have a National Silk Consortium having the complete genome of the silkworm. So we can understand through this particular lesson that bivoltant, univoltant races, pure ones, produce high quality of good, high quantity of good quality silk. Multivoltant or polyvoltant races are going to be tolerant to pathogen loads. They are going to be hardy, resistant to different kinds of diseases. But what they do is they are going to produce low amount of silk that is poor in quality. And egg chorion color, you can visually discriminate it on the three replicates, and the color is either white, yellow or mixed. Cocoon size, randomly around 100 cocoons are taken and the sizes can be determined big, medium or small. Population rate, I'm sorry, it is pupation rate. When you see it in percentage, It is number of the cocoons having the pupa which is alive. So life cycle of the silkworm, you have egg, larva, pupa, and the adult. So the pupation rate is wherein, the number of cocoons that are present having alive pupa by the total number of larvae that are counted into 100. We are talking about percentage. Fresh cocoon yield by one box of eggs, which is around 20,000 eggs is in 1 kilo, is going to be calculated by fresh cocoon yield by laying per replicate in kilograms into 20,000 into hatchability into zero point 0.1. That is your hatchability percentage by the number of larvae that have been completely counted. Your filament weight in grams is the one found after the cocoon is reeled, the filament is dried, and that dried constant weight to the weight is being measured. Most of the silkworms, they're going to be having a high hatchability, and 80% of the silkworms, they have accessions having pupation rate higher than 85%. Some 26% have the pupation rate, that is formation of the pupa from the total number of larvae counted. It's around 85 to 90%. 39% of the complete silkworm breeds, you have around 90 to 95% pupation rates, but it's only 15%. The top breeds manifestation show that the pupation rate is higher than that of the 95%. So you have less amount of wastage. So again, genetic characterization of the silkworm gene pool at the molecular level is also checked. You have different kinds of markers, AFLP, RAPD, different kinds of markers which will tell you and regarding the DNA of the particular silkworm gene. This, after you perform the what do you call it, your micro injection, and then you do your PCR analysis, you can find out whether the hybrid is formed or not. So this is all about your second topic. Now I would like to start with the hybrid vigor and heterosis as well. In this, we are going to see what exactly is going to be heterosis, how important it is, and what exactly is the hybrid vigor. So heterosis, hybrid vigor, outbreeding and is the improved or the increased function of any biological quality. So it is all going to be how good are your genetic traits? Is there any kind of improvement? So an offspring is the heterophyte, and if the traits are better than the parents, this is what is hybrid vigor or heterosis. So genetic contribution of the parents has been. Kaustubh, I need my phone. Kaustubh, phone. Phone. Phone is outside. Give me the phone, Anna. I do not want. నాకు ఇలా చేస్తే తన్నేస్తాను కౌస్తుభ్, ఎస్పెషల్లీ డ్యూరింగ్ క్లాస్ టైం. ఎప్పుడన్నా ఆడు కానీ ఇప్పుడు నాట్ వెన్ ఐ హ్యావ్ క్లాస్. సార్ క్లాస్ ఎండ్ చేశారా లేకపోతే కనెక్షన్ పోయిందా ఏంటి? ఓకే. ఎందుకంటే మాకు 10 చాప్టర్స్ ఇచ్చి 6 క్లాసెస్ ఇచ్చారు. సో మేము యాక్చువల్లీ 1.5 క్లాసెస్ పర్ సెషన్ అయినా తీసుకోవాలి. అది చూస్తున్నాను నేను. ఆల్రెడీ 5 మినిట్స్ ముందర పోయింది, మళ్ళీ ఇప్పుడు 5 మినిట్స్ ముందరే ఆపేశారు. సో ఐఎమ్ అవుట్ ఆఫ్ 10 మినిట్స్. అది నేను చూస్తున్నాను ఇక్కడ. ఓకే. ఓకే. ఓకే సార్. ఇంకా యూట్యూబ్ మీటింగ్ ఇస్ స్ట్రీమింగ్ లైవ్ అని వస్తుంది. ఆ, ఓకే సార్. నాది కూడా ఇప్పుడు లాస్ట్ ఇది రికార్డ్ అయింది. ఐ థింక్ కెన్ యూ జస్ట్